What if genetically modified organisms could not only revolutionize how we farm, but also protect the environment? From drought-resistant crops to bacteria that produce life-saving medicines, genetics research has transformed the way we interact with the world we live in. A future where GMOs are the key to solving some of the biggest environmental challenges might not be too far off. Scientists could genetically modify animals to reduce agricultural pollution, opening opportunities for a revolution in sustainable farming. By creating genetically engineered pigs to produce low phosphorus manure, they are attempting to dramatically reduce runoff water pollution. Could these innovations be the key to a better, cleaner future? Welcome to the genetic basis of stuff and things. The main priority of genetically modified organisms throughout agricultural history has been to increase productivity. For example, Roundup-ready crops are crops engineered for herbicide resistance, and Bt corn is pest resistant. Even the first genetically modified animal to be FDA approved was Aqua Advantage salmon, a breed which could mature and grow faster, increasing efficiency of salmon farms. Boosting agricultural production is important, but what if genetic engineering technology could be used to solve one of the greatest challenges faced in the agricultural industry: pollution? Between 2002 to 2010, agricultural practices contributed to 38% of the phosphorus load in global freshwater systems. Phosphorus runoff from agriculture is one of the leading causes of nutrient pollution, leading to eutrophication of bodies of water. This results in hypoxic environments, which leads to excessive algal blooms, death of aquatic animals, and disruption of food webs. Phosphorus is an essential nutrient for pigs, playing a vital role in bone formation and overall metabolism. However, there's a problem. Naturally, pigs cannot efficiently digest a substance called phytate, which is the primary form of phosphorus in most plant-based feeds. Phytate binds to available phosphorus, rendering it indigestible to pigs. As a result, the pig's digestive system is not able to absorb the phosphorus, meaning most ends up in the pig's manure. In the livestock industry, this leads to overuse of phosphorus supplementation or adding enzymes which help the pigs to digest phytate to the feed to ensure pigs meet their nutritional needs. However, these specialty feeds rely on diminishing supplies of global phosphate reserves. Rather than engineering the pig feed, what if you could engineer the pigs themselves to digest plant phytate more effectively? That is exactly what was achieved in a groundbreaking 2001 study by Sergey Golovin and colleagues. The innovative solution proposed was Enviropigs. Enviropigs are genetically modified pigs which express an enzyme called phytase in their saliva. Phytase breaks down plant phytate into usable phosphorus, making it biologically available for the pig. By inserting a bacterial phytase gene into the pig's genome before birth, the pigs were able to express this enzyme in their salivary glands. So, as the pig chewed its feed, the phytase would already start breaking down phytate in the mouth. So, how did they do it? To achieve the expression of the phytase gene, the team undertook a transgenic modification of fertilized pig embryos. First, they isolated the phytase gene from the Escherichia coli genome. They then cloned this gene with a promoter region, the pig parotid secretory protein, which ensured specific expression of the enzyme in the pig's salivary glands. The transgene was directly injected into fertilized embryos, which were implanted into surrogate mothers. The baby Enviropigs were then verified and bred to produce stable lines of organisms which contain the transgene in their genome. The results were impressive. The Enviropigs, which express salivary phytase, absorb significantly more phosphorus from their feed compared to non-genetically modified pigs. For non-transgenic pigs, the digestibility of phosphorus was about 50%. Contrastingly, the phosphorus digestibility approached 100% for the transgenic pig lines. This meant that Enviropig manure contained up to 75% less phosphorus than regular pig manure. Reducing the phosphorus content in agricultural waste by such a large margin has enormous implications for the environment. It could greatly reduce the impact of food production on water pollution, allowing farmers to operate more sustainably. As a study which pioneered the production of GM animals for sustainable farming, this was also a trigger for important discussions about the ethics and safety of genetically modified livestock for human consumption. The Enviropigs developed in this study displayed normal health, growth, and behavior just like their non-GM counterparts. However, introducing GM animals into the food chain is a topic that is heavily regulated. Public concerns have limited Enviropigs from being widely adopted in agricultural practice. In 2015, a huge step forward was made with the FDA approval of the first genetically engineered animal for commercial use, Aqua Advantage salmon. However, we still have a long way to go until genetic engineering of animals is universally embraced. By drastically reducing phosphorus pollution, Enviropigs could help protect the environment while keeping agriculture productive. But the road to widespread adoption of this technology is regulated by debates over ethics and safety. As GMOs continue to evolve, they might just be the key to a cleaner, greener future if we're ready to embrace the change.